Okay, good afternoon, everyone. So welcome to the Theoretical and Computational Biophysics seminar this week. So we are hosting Dr. Mohsen Sadeghi from Free University of Berlin, Department of Mathematics and Computer Science today. By way of introduction, Mohsen received his bachelor's, master's, and PhD degrees from mechanical engineering at Sharif University in Tehran, which is the best technical university of the whole region, not just the country. And then, even though he was offered actually an appointed faculty position as an assistant professor at Tehran University, he declined that offer and decided to join as a postdoc to the group of Frank Noe, whom you know, in Berlin, at the Freie Universität in Berlin. And since then, actually, overall, during his tenure, he has been working on many different computational physics and simulation-related problems. And recently, he got interested in membranes, apparently, which is very interesting for us. And we would like to learn a little bit more about his approach to membranes and how to describe them at ultra-coarse-grained levels, realistic kinetics of coarse-grained models of lipids, and actually description of proteins in membranes, which is very interesting for many of you who are looking at aggregation of proteins and membrane on the surface of the membrane or inside the membrane and so on and so forth. So he was kind enough to accept my invitation to give us a seminar, even though it's late in Berlin, so it's late evening, but he agreed to give us a seminar at a standard time. And separate, we discuss, and actually Mohsen is available to talk with you after the seminar. So if you want to stay on, we can use the same Zoom link. Thank you very much, Mohsen, for being here. So we look forward to your presentation. Please.
Thanks for the very nice introduction, and thanks for having me. I hope I can give you a kind of a general overview of what I'm doing with this so-called dynamic modeling framework. And without further ado, let's jump in. As a means of introduction, I guess you know a lot about biomembranes, these self-assembled structures of phospholipids in the form of bilayers, the usual story with the hydrophobic, hydrophilic parts. And what's interesting is that from a biophysical point of view, the biomembranes or membranes in general, they can have a variety of morphologies, topologies, even changing shapes, scission, like membranes fusing and ceasing, and you can have interesting phenomena happening from this point of view. For example, this is rather recent work in which the system of mean proteins on the membrane introduces oscillations in the neck and sometimes breaking the neck. And from a purely mechanical point of view, membranes are kind of interesting because they are rather rigid structures. You need energies of the orders of tens of kT to bend the membrane. But on the other hand, there are fluids in planes, so stuff can easily diffuse around in the membrane. You can have free diffusion in 2D, but the whole structure has a rather large rigidity. So it's interesting to capture this basically combination of different properties in one model and to see what comes out. One other aspect is that kinetics can be rather important in membrane phenomena, and this is something that is less well understood or well studied. As a general example to see what I mean, this is the study on the retrieval of synaptic vesicles. So these membrane vesicles, basically, the task is to retrieve the neurotransmitter from the synapse junction. And people are basically still talking about what is the actual mechanism that this happens. It could be very fast, so that the pre-made vesicle can come join with the membrane, get the neurotransmitter, or it could be that the collagen-mediated endocytosis is in play. And it all boils down on how fast this happens. And when talking about membrane proteins, we might refer to different things, and let's get some basically naming conventions out of the way. So we might talk about integral membrane proteins, proteins that are embedded inside the membrane. Examples are, for example, channels. Or we could talk about peripheral membrane proteins that sit on the top of the membrane, bind to the top of the membrane, and have some function related to that. There could be also proteins that are more weakly anchored on the membrane, and they do not take part in mechanics of the membrane or changing the shape of the membrane, but could be acting as receptors, for example. Focusing on peripheral proteins that tightly bind to the membrane, they're usually lipid anchored. They can induce curvature in the membrane with variety of reasons. Could be helix insertion, could be the mismatch on the binding sites. And they are majorly responsible for membrane remodeling. So you remember, membrane is a rather stiff structure. You would need to expend energy to deform it in any shape. And you need to have a scaffold structure that would keep the membrane in a certain shape. So for example, this is a cryo-EM image of part of ER, the endoplasmic reticulum. And you can see these worm-shaped things. These are bar domain proteins that are all over the structure just to keep it into a certain geometry. One good example of these peripheral proteins is the shigatoxin, the so-called shigatoxin. It's a protein with two subunits, the so-called subunit A and B. The function of subunit A is to kill you, so it's toxin. The function of subunit B is to get the subunit A inside the cell, and this is more interesting. So as I will mention, this has a very elegant entry mechanism, which people are now looking into to, for example, attach another cargo, a non-toxic cargo, for example, an antigen, and then when you leave it in the body, the subunit B will take care of getting the antigen inside the cell. And you can use it, for example, for vaccine delivery or drug delivery in general. The interesting aspect of this is that people have, for example, left the subunit B on a GUV, on a giant unilamellar vesicle. And so this is just a bubble of pure membrane. It has no internal machinery involved. Still, this protein manages to build long tubules into the cell. And it has been suggested that this membrane curving protein can cluster on the surface of the membrane, form a large pit, and these pits could evolve into invaginations and long tubules. And when this happens, so when rather a small neck is formed into the cell has the machinery involving dynamin that would assemble on the neck and basically seize the neck. And so this is what happens when the toxin acts in your body. And it's interesting from a membrane mechanical point of view, how this passive entry mechanism actually works. If we want to do simulations on this system, we might look at a range of scales. So you could have all atom models of proteins, the scaffolding on the membrane, which would limit your range of study to something like tens of nanometers. You could have coarse grain models in the sense that you still have representations of lipids, individual lipids, but with beads that include a cluster of atoms. Or you can go all the way across the scale to where the membrane is basically represented by beads that do not directly represent lipids or do not directly correspond to individual lipids, but still reproduce an overall mechanics expected from membrane. Then you could have different representations of these proteins in there as well. With this type of models, we could push the limits to larger scales. But the important question is, as we move across this ladder of scales, how not to lose information on protein flexibility, its lateral dynamics on the membrane, and ideally membrane-mediated interaction, as I will talk about a bit more. So this is the membrane model that I had been working on for the past few years. This is a model which is formed by an assembly of particle dimers. So one particle per leaflet of the membrane. And these particles interact with their nearest neighbors. So there are bonded interactions involved between the particles in plane, also angle bending potentials resisting the rotation of these bonds out of plane. And in general, I use a top-down coarse graining to parameterize the model. I'll talk about that. It involves a so-called bond flipping moves to allow for in-plane fluidity. I will also mention that. And a bit about how I integrate this. To give you a sense of the scale, I have done this rendering in the bottom of the slide that you can see. This is an actual Shiga toxin sitting on top of this membrane model. Everything is up to scale. So one bead of the model is comparable in size to the whole protein. So everything needs to be encoded into how this one bead interacts with its neighbors. So how do I go about parameterizing the force field?
Sorry, Mohsen, how many lipids fit into one of your beads?
So it depends on the size of the bead is a controllable parameter. Could be something between 20 lipids up to lipids,
Okay, but you do have two leaflets, right?
I do have two leaflets.
Okay,
yes.
okay, got you. Thank you.
And this is the general parameterization scheme. On the one hand, you have the particle-based model, and on the other, because I'm interested in the top-down parameterization, I have a continuum representation of the membrane. And we can connect the geometry of the two models through some differential geometry. And we can also connect the energetics of the two descriptions. So on the one hand, we have some curvature elasticity representing a continuum model. On the other hand, we have these force fields and interactions. And ideally, we would have a way of having an energy equivalence between the two descriptions. So in the force field part, I have, for example, here a Morse type interaction for bond stretching and a usual harmonic angle bending. And these two would basically be summed up to give me an effective energy density on the surface of the membrane. On the continuum side, we have the well-known Helfrich functional, which gives you the energy density per surface of the membrane based on the mean and Gaussian curvature. And it has two parameters, a kappa and kappa bar. These are elastic constants that you can measure. I have additionally included a function that I will talk a bit about, which includes the elastic contribution and all the other effects that the protein would have on this model. And this whole thing would go into an optimization procedure. And this optimization procedure would give me, so looking to the left, you see here two cases of particles, the white one and the red one. The white one is where we have pure membrane, and we have the Helfrich functional. And these wireframe plots are the energy as a function, the energy density in the landscape of curvatures. So I can hypothetically bend the membrane and expect the energy that comes from the force field to match the energy expected from the macroscopic description. On the red particles, we additionally have the protein, which in this case affects twofold. It introduces a preferred curvature. It has an intrinsic curvature. Also, it will change the elastic behavior of the membrane. So we have a bit of a tilted and shifted energy landscape. We can still parameterize for. On the right side, you see the effect of this bond flipping moves. So if you consider two triangles in this bonded topology on the surface of the membrane, the shared bond between the two triangles can optionally be flipped to the other side, to the other diagonal. This is a standard way of introducing fluidity into these bonded models. And we can also control, for example, the probability of this happening. So in the end, this is a Monte Carlo move. When a move is suggested, it has to go through the Metropolis-Hastings algorithm to be accepted or rejected. So with this, we can control the in-plane viscosity of the membrane. And this is an example in the video. This is a membrane that is flowing in plane under a gravity-like force, so to say. And this is another example which includes several of these factors in one model. So now I have spherical nanoparticles interacting with the surface of the membrane as modeled with this force field that I described. And these nanoparticles basically might be wrapped completely by the membrane or be basically left with the partial wrapping on the surface of the membrane. This very much depends on the ratio between the adhesion forces and then the rigidity of the membrane. So this small u parameter. People have looked at this from a theoretical point of view. There is a thresholding behavior around the value of the u value of 2. And I could reproduce the same behavior using this particle-based model, which basically tells me that the underlying mechanics of the membrane and these interactions are playing together nicely. Sorry, was there a question?
Yeah. So your membrane doesn't rupture, right?
No, here it doesn't rupture, no.
Okay. Even though you're using a Morse potential for the
right.
bonds.
So that's right. Ideally, it can. So this is kind of one reason that I use the Morse potential to see where I exceed the tension that would rupture the membrane.
But from the video, I see that if you can
doesn't happen here.
Right? Yeah. Okay. Thanks.
And for example, I also checked the shape of the neck that is formed around the area that the membrane buds and engulfs the nanoparticle. Ideally, it should be a catenoidal shape. And this green curve is the catenoidal fit to the shape of the membrane. So the geometry, reproduced geometries also makes sense. And this was a nice example in terms of the more or less the outputs. And the people from the Allen Institute, they were building a software called Simularium, which I encourage you to look at. This is an interesting web application for visualizing simulation outputs. And they used my nanoparticle wrapping simulation as an example.
We were just there last week at the Allen Institute.
Right?
We were just there last week, actually.
Ah, very nice.
Interesting.
So you know this people.
Yeah. Okay.
Interesting. So aside from, I talked a bit about the in-plane viscosity of the membrane, which is a kinetic property, but I got a bit interested into reproducing the membrane kinetics, especially when we have the solvent. So most of these coarse-grained models, they work in a vacuum, so to say. So they are so-called solvent-free models. And then you basically cannot trust the undulation kinetics of the membrane, the out-of-plane kinetics, how fast the membrane would develop these modes. And if we have an example in which this comes into play, then we are in a bit of a jam. So how can we fix that? And I came up with a as simple as possible way of including solvent kinetics into this model. And this works like this, that I introduce a test boundary condition, a test displacement or force on one of the particles, and then solve the Stokes equations for this Gaussian-like perturbance in the displacement or force of this particle. The resulting fields of the fluid, which could be the displacement field or basically the velocity field or the stress field, could be integrated on other particles, and that could give me a linear response model of the fluid. I could incorporate this into Langevin dynamics, and this would give me reliable kinetics. And optionally, I could just use it per particle. So one particle has an effect has an interplay with the solvent. It could affect its nearest neighbors, or it could affect the whole particles in the model. And so with this now enhanced kinetic model, now I can look again at the kinetic properties as reproduced by this model. First, let's also take the in-plane kinetics out of the way, comparing the diffusion coefficients that I would get for the peripheral proteins modeled as I described. You would have values very much comparable. So this plot to the right, this comes out of the simulation. So these are mean square displacement of these proteins diffusing around on the membrane, and down there you see the diffusion coefficients. And these values here are actual experimental values measured for the diffusion of these proteins. So this model very nicely reproduces this very slow diffusion. So these are proteins that bind with multiple binding sites to the membrane, and they diffuse laterally very slowly. Also, the overall surface viscosity of the membrane can be verified with the experimental values. So by looking at the fluctuations in the shear stress, I can come up with effective values of membrane surface viscosity and compare it with what people have measured in experiments. So still, this is a rather wide range experimentally, but interestingly, the values that I measure also fall into the middle of this range, more or less. So we have a membrane which has a, yeah, go ahead.
So how is the protein represented in these simulations actually? So then, is it a coarse grain protein? Is it what, just one bead?
Let me see if I included that. No, a bit further forward, but I will give the general description.
Okay.
Yeah. So let's finish up with the kinetics. And now to verify the out-of-plane kinetics, I've compared the dynamics of these fluctuation modes with a well-known model, which is called the Seifert-Langer model. It gives you the dispersion relations of the membrane, so to say. So what are dispersion relations are the relation between spatial and temporal frequency in a general model. And for the membrane, it means that each of these fluctuation modes, which could have a spatial wavelength, how fast they would develop or dissipate. And this is something that I can measure and quantify in the simulation as well. Interestingly, they match the prediction of this Seifert-Langer model very well when I included the hydrodynamic interactions across the membrane. Also, the model reproduces the equilibrium thermodynamics behavior nicely. So apart from the kinetics, the underlying thermodynamics is also unaffected. This is the famous 1/Q4 relation that you get for thermal undulations in a membrane. Yeah, as I mentioned, if I reduce the range of hydrodynamic interactions, it damages the dispersion relations predicted by the model. And as a non-equilibrium case, which would put the spotlight on the kinetics, I looked at the system of bacteriorhodopsin. This is a transmembrane protein, which is activated by absorbing photons. This is a proton pump. It can push one proton across the membrane actively. And in doing so, it exerts a small amount of force on the membrane. These form the so-called purple membranes, if I'm not mistaken. And people look at this, you can shine light on the membrane with these proteins and watch it be actively agitated, so to say. And there was a good theoretical description of this system with these proteins we described as basically particles on the model, which could have one of three states, being off or pushing up or down on the membrane. And people looked at this and measured what would be the mean square displacement in terms of the height fluctuations of the membrane resulting from the activity of this. And I mean, this is a large formula, but interesting part of it is that this average property strongly depends on the viscosity of the solvent, the eta, as well as the lateral diffusion coefficients of the proteins. So kinetic properties go into a measured average property, which is basically a hallmark of having non-equilibrium steady state. And down here, you see a plot of the model reproducing exactly what was expected from this theoretical prediction. The dashed lines are the predictions of the model, and the solid lines point to where this model predicts. And furthermore, we can see that these fluctuation modes no longer follow a Gaussian distribution when we have active contributions. So there's a certain non-Gaussianity, so to say, resulting from that. Now let's talk a bit about the actual presentation of these proteins, as I mentioned. So this is now focused on this shigatoxin system. I'm looking at this protein as more or less an elastic being that sits on top of the membrane. It has its own elastic model. I've arbitrarily chosen the Kirchhoff-Love small deformation theory. This has something to do with my mechanical engineering background, so this is something that I understand quite well. That what would be the resulting energy of bending an elastic plate. And I'm modeling the protein as being composed of such an elastic object. Now we need to put in some mechanical properties into the protein, more specifically a Young's modulus, which gives you a stiffness, so that how hard it is to deform the protein as would be in this case. And this is something that people can also measure, for example, using AFM tips. You can push on the protein and then see what's the resulting displacement or deformation. And these are usual values that people have measured. So, for example, for capsids or for filaments, could be rather large. And so these are the stiff structures. As you go, for example, to this antibody, this gives you a very small Young's modulus. In the model that I built for this protein, if we have a stiffness of about 35 megapascal, we would have something which is as rigid as the membrane. So I would expect these peripheral proteins to be upwards of this value in terms of stiffness so that they can imprint on the membrane. And let's see, okay, this is a simulation of this system. This is basically the trajectory of these shigatoxin-like proteins sitting on the membrane and diffusing around. And the way that each of them affects the membrane is that there is a force field masking procedure happening. So as this particle diffuses around, it carries with itself a certain substitute force field or a certain additional force field contributions that are that they also go through a Monte Carlo move to be accepted or rejected, and they can locally change the properties of the membrane. And you can see that in the end, these guys aggregate on the surface of the membrane without any direct interactions between protein particles. So whatever is happening is membrane-mediated. It has to do with the mechanics of the membrane. I measured the effective curvatures that come out of this simulation. So this is partly something that is presented in the parameterization step, but I can also look at it after the simulation, see how good it is reproduced, so to say. So you can compare the arrows with the dashed lines, and the arrows are what was dictated when we were parameterizing the model, and the dashed lines are what is averagely produced by the model in the simulation. It's a bit lower in each case because of these Monte Carlo moves. So not always proteins are successful in dictating the curvature that they like. And this range of curvatures are also not fictional. So comparing with MD simulations of a shigatoxin, you can see that this would correspond to somehow mid-range curvature that I have in the model. But for a chlorotoxin, people have done experiments with curvature sorting. So they put the membrane on an already curvy substrate and see where the proteins would accumulate, and this gives them the preferred underlying curvature. So these values would be the higher range in the curvatures that I can reproduce. So I wanted to look at these membrane-mediated interactions in a bit more detail. So I came up with this 2D fluid mixture model. So let's say that looking from the top, we have membrane particles and we have protein particles and we have all types of pairwise interactions. So now these are inferred interactions. These are not interactions present in the model. I'm trying to fit a theoretical fluid model into what I observe in the simulation. So as a first step, you could use the Kirkwood theory. You look at basically the pair correlation functions and measure a potential of the mean force. This is an easy procedure. The problem with it is that when I measure, for example, membrane-membrane and membrane-protein interactions, I can no longer subtract them. I can no longer manipulate them algebraically because these are now free energies, and they came up with, so it's not easy, or it's not straightforward to add or subtract free energies in this case. And yeah, these are like more explicitly mentioned what I'm talking about. So to do it more directly, I moved a step forward. So this is the application of a direct correlation function. You can find out this C alpha gamma function. So between any pair of entity in your model from the pair correlation functions. And this is the so-called Ornstein-Zernike relation between the H and C. H is just comes from the pair correlation function. This is something I can measure, but what good is it? You can put this direct correlation values into a closure relation. So you can relate them to the actual interaction potential between the fluid particles. And this is something that I did. And then I could easily subtract the resulting potential. So now I can subtract the membrane-membrane interactions from protein-protein interactions to come up with the pure membrane-mediated effect. And these are the results that I measure. So they are, in the case of the stiffest protein, a bit larger than 1 kT in terms of the potential well. And I could also study the effect of the protein stiffness or its surface concentration. The fact that the interaction potential or interaction strength depends a bit on the surface concentration tells me that there could be multi-body effects present in the system as well. And in terms of what's happening, so what's the underlying mechanism of this membrane-mediated interactions? Generally, people categorize this into two different types of mechanism or two different origins of the forces developed in the system. There could be enthalpic and entropic. So if you have two membranes or two species basically bending the membrane, so if you have two proteins or two other species bending the membrane, you would generally expect repulsion happening between two. And you can think of it as something similar to what happens in general relativity. So people in membrane community are very humble people. They basically borrow things from this high-end physics theory. So with the entropic force is even worse. So this is if you have entities rigid enough to suppress the membrane fluctuation, people call it the thermal Casimir effect. There would be an attraction resulting from the entropy of this fluctuation mode. So apparently the membrane would like the inclusions or the peripheral proteins to be put together to allow for more fluctuation modes basically be liberated. And this would be an entropic effect. It manifests as an effective attraction between the two.
Sorry, Mohsen, why do you think they repel each other, the first part? I didn't quite understand.
Right.
What?
So, right. The intuition behind it is that if you consider only one inclusion, you come up with a certain shape of the membrane as the zero energy configuration. And if you put another configuration on another protein on top of it, it would perturb this. And so the minimum energy, if you calculate basically the interaction between these two profiles, the minimum energy would only be achieved if they are infinitely apart. So there are no zero energy configuration. Yeah, that's why.
Because I ask because we know there are proteins that prefer certain curvature in a membrane, so they accumulate in the regions of the cell where you have that type of curvature. And I was kind of intuitively not agreeing with the model that you're describing here. Okay,
yes.
I see.
No, you're absolutely right. The story is a bit more complicated than what I put here. Yes, yeah, definitely. If there are preferred curvature areas, or if you have multi-body effects, or if you have basically anisotropic curvature. So this is a case where the curvature induced is basically rotational isotropic. If they're
see.
anisotropic, they can find configurations.
Oh, interesting,
Yeah.
interesting. Okay, thank you.
You're welcome. And so I looked at the aggregation behavior from a coagulation or polymerization point of view. So let's see what's the dynamics of these protein clusters forming or dispersing. And I found a nice model which looks at this process in terms of a reversible reaction happening between two clusters of size I and J coming together, forming a larger cluster, or breaking apart, forming the initial ones. And it's a kinetic model in the sense that basically the formation or fragmentation rates can depend on the cluster size. Interestingly, with certain assumptions which I have omitted here, you can look into the paper for a detailed description of this. We could find the stationary distribution corresponding to this model. What would be the stationary particle size, basically, formed by these proteins coming together? There is a nice expression in this with a mu prime parameter, which looks like a chemical potential. So now this is kind of a chemical potential of particles joining or moving between clusters. Also, this model allows for the formation of one large cluster, which is basically a sink for particles. So if a particle joins this large macroscopic cluster, it gets out of the system. So there is a G star parameter, which tells you in the end, in the equilibrated case, which fraction of particles have joined this macroscopic cluster. I took the simulation trajectories. I used this DBS scan algorithm to detect clusters in that and find what's the distribution of cluster sizes and come up with the actual values of these parameters. This model fits the observed cluster distributions quite nicely. We could measure the chemical potential, which are on the order of kT per particle, which is comparable to the effective interaction that I measured with the other fluid mixture model. And interestingly, I found out that this paper was later cited by a couple of people who were doing experiments, and they can detect the size of these clusters. The same model apparently worked for them as well. Furthermore, I tried to do some free energy analysis on this system, and I came up with this rather weird reaction coordinate or collective variable. So I'm using the mean of inverse of pairwise distances between proteins as a reaction coordinate. So you can assume that if a lot of them are together, there are small pairwise distances, and the inverse would give you a large value of Q. If they are completely dispersed, you have vanishing values of Q. And I could see that, for example, if I change, so I selected the snapshots from the simulation in which the mean cluster size only differed very small in value, and I could see that the distribution of Q values are completely separated, completely bimodal for this. So it's a good discriminator of the state of cluster formation on the membrane. And to obtain a free energy landscape or free energy profile along Q, I basically made an assumption that because of the fact that I'm using the force field masking to include these proteins, each of these simulations correspond to a biased simulation, with the bias being correlated to the strength of bias being correlated to the density of this particle presented in the model. Then, if this assumption holds, which I verify by seeing if the biasing strength correlates with the surface concentration, then I can put this in a basically standard free energy estimation method and combine simulations with different surface concentrations to come up with the free energy landscapes. This is what I did. And yeah, these are plots a lot along the Q and the gamma as the surface concentration. More interestingly, if we take the mean free energy as a function of the surface concentration, we can see this similar curve. This is the classical nucleation behavior that you would have, that free energy increasing up to a certain concentration, then afterwards it becomes favorable to form clusters on the membrane in this case. So the free energy would go down with concentration after that. And the derivative of that would give us again, a chemical potential. And the change, basically the difference between the two chemical potential values, again, is similar to the KT value that I measured previously. Also now with this specific form of bias, I can look at the effect of protein stiffness in my model. So how would the free energy change if I change continuously the protein stiffness? Interestingly, again, I get a double behavior. So up to a certain stiffness, actually the system doesn't like when you make the proteins more rigid. Apparently, we have these repulsive interactions up to this point. And after a certain value of stiffness is reached, then attraction dominated behavior comes into play. So there would be now preferred behavior as having proteins coming together. And so when I have this free energy estimation method in place, I could look at more interesting stuff. In this case, I looked at the entropy of the whole system as a function of, again, this reaction coordinate. So how does the entropy change as I have more aggregated or dispersed cases? Due to comments from a reviewer, I switched methods in estimating free energy. Now I used a method that was actually developed in our group, but it was a bit more complicated than what I used before. So it's called the transition-based reweighting analysis method, or TRAM. This is a very nice method that can look at even out of equilibrium trajectories or partial sampling of a system without having the full equilibrium trajectory, and build an underlying Markov state model and estimate the free energy differences for you. And using this, I could very nicely calculate the entropy of the system in very continuous curves as a function of surface concentration. So in this plot down left, you can see the entropy as a function of Q for 4 different surface concentrations, starting from the gray and blue curves, moving to the red and green one as its concentration increases. Interestingly, for low concentration cases, we have the entropy increasing with Q. So it would result in, so we have entropic forces that would push the proteins together. But for highly concentrated cases, it actually damages the overall entropy, which is to be expected. So if you have a dense system, you're damaging the configurational entropy of these particles when you want to cluster them around. And this is something that very nicely can be compared in this model. So overall entropy of the whole system, I could use the combinatorial estimate of the configuration of proteins on the membrane and compared with the entropy that I estimate from this model. The behavior more or less compares, and you can argue based on where this goes over or below the expected combination or combinatorial entropy. And finally, I also did a more or less a direct estimation of entropy by looking at the distribution of modes, as in undulation mode of the membrane, as well as orientational entropy of a director field, that because of the fact that I have 2 particles per membrane, so I have 2 leaflets, I can also estimate the local director field and look at the entropy of that. I could reproduce the same behavior that for low concentration, the entropy, even with this very qualitative approach, still the entropy increases with Q when the surface concentration is small, but as you go towards denser systems, the entropy prefers more dispersed states. And so this is the story as to the entropic forces. And with that, I'm done with the general presentation. I do have a promotional message here. We are editing a topic for the Frontiers in Physics journal with Thomas Weikl and a few other friends of mine. And this is still accepting articles. So the official deadline was until yesterday, but we are extending that. So if you have something along the lines of this organization, aggregation, and self-assembly at membranes, you can submit to this issue. And thanks for your attention. These are two other movies for your entertainment. So the one to the left is something that I'm working on. This is a steered simulation in which I force the particles to come together, and this results in turbulation of the membrane. And the one to the right are my attempt at modeling bar domain proteins. So now these are anisotropically curving proteins, and they would result in vesicle formation. Thanks again. I will take your questions at this point.
Thank you very much, Mohsen. Very, very interesting. I have a lot of technical questions, but I've been asking you questions already. Let me see if there are any questions among the students we can start with. And then I can ask more questions.
Sure.
Any questions for Mohsen, model and application from the audience? You can raise your hand or turn on your camera and start asking your question yourself, or type them in the chat.
Can I ask a question, Emma?
Yes, Safar, go ahead.
Thank you very much, Mohsen. It was a great talk. And I was wondering, so some of them integral proteins that we have in the bilayer, they don't exactly deform the membrane or curve the membrane. They basically thicken or thinning the membrane.
Right.
So does your model capable of, predicting these kind of behaviors?
Of course. I mean, that's kind of the major reason that I'm trying to reach out to other groups doing other stuff, because as long as this is an effect that we can incorporate into the force field at the scale that the model works, this is reproducible. So we can play with the thickness of the membrane, as you said. We can have basically different local rigidity. We could also manipulate the local fluidity. So if these things affect the viscosity in the vicinity, we can also incorporate that into the model. Sure.
Thank you.
Interesting. So I didn't quite get the membrane protein interaction. Is there something that you put into the model, a kind of force field for the interaction between lipids and protein?
Right.
And how do you do that?
Okay. So the way, at least for this Shiga toxin system that I described here, the way that it works is that there is an additional set of interactions. So a particle that hosts a protein would have additionally different interactions with its neighbors. So there is a local curvature incorporated into these interactions, as well as a change in the stiffness. And these are all parameterized consistently in the same procedure. So the value for these additional parameters also would result in the same energy landscape as I put in. And then when this particle diffuses around, it carries this set of new parameters. So I call it
I see. So when you look at the diffusion of the protein, for example, so what is it? What is the main parameter or factor that determines the diffusive behavior of the protein?
Right. So in this case, because of the kinetic model that I'm using, there are two parameters that work in tandem. One of them is a general viscosity that I can locally attribute to the particles that move around, as well as the frequency of proposing these bond flipping moves. So this is an additional parameter that we can manipulate.
I see. And then I had one question about your bacterial reduction case. So the peripheral membrane is a very dense membrane. I think you have 75–80% protein, and a little bit lipids sitting between the proteins. So you mentioned there are experiments, so I'm right.
Yes.
So these experiments are done with the peripheral membrane, or is it with isolated bacterial reduction reconstituted in a membrane like what you're showing in your figure?
Right. That's a great question. So if you look at my figure, this seems like a very dispersed system with like active protein. And so the experiments are usually done with that dense assembly that you mentioned, because the individual magnitude of these forces are very small. So I kind of cheated in the sense that now these are basically islands, including a lot of these bacteriorhodopsins, but the islands are now dispersed. Yeah.
Gotcha. Yeah, sure. I see. Very interesting. I had no idea that bacteriorhodopsin during its function can affect the membrane. I mean, from my perspective, it was a very rigid proton pump sitting there and just doing something in the middle. I didn't expect it to kick the membrane essentially while it's pumping protons. Very interesting. I had no idea. Okay, so then one last question. So this is implemented in your own simulation kind of engine, or is that right? Okay.
Yeah, everything is a specific purpose software. Yeah.
So we have to work with you if you want to use your method.
I come attached. Yeah.
Fair enough. Yeah, very interesting. Very good.
Thank you.
Okay. Any other questions, guys? If not, let's thank Mohsen one more time. And then Sefer, you can stay on to interact with Mohsen. So I guess we're going to discuss actually the model that you have, Mohsen, with the group, and then see how we might be able to use it for our protein cases and system. We have a couple of cases that actually we are looking at aggregation of proteins on the surface of the membrane. And it would be great if you could actually take a look and see what we get from your model. Like you said, how we can improve your model, or we can get long-term scale behavior. So this is corresponding to how many microseconds? What you can capture?
These are millisecond simulations.
Oh, so, no, millisecond. Wow.
Yeah,
perfect.
So I can easily do millisecond simulation.
Great. And then any protein we want to use, we have to kind of train your model with that particular protein, shape and mechanics, and then we can put, okay, got it.
Yep.
Very good. Thank you very much again. So you can stay on if you're not too tired.
No, that's fine.
Chat a little bit with Sefer, who wanted to talk to you. Chun, you have anything to ask? I see your camera is on. Okay.